Nagy szeretettel köszöntök ismét mindenkit. A Magyar Tudományos Akadémia Biofizikai Bizottsága, valamint a Magyar Biofizikai Társaság, és hát a magyar biofizikusok nevében ezen a mai összejövetelen, aminek rövidített címe Békésy Symposium, és egy nagyon neves előadógárdát sikerült ehhez összeszednünk, és az ő előadásaik alapján szeretnénk megérteni, hogy mi történt Békésy György munkája óta a hallás biológia terén. Mivel nagyon sok külföldi előadó van, ezért most angolra fogok váltani, mert őket is köszöntöm. Dear colleagues, dear audience, dear speakers, it's a great honor and privilege to welcome you on this evening or this afternoon or morning in the United States, and we are about to start the Békésy Symposium, and I would like to welcome you on behalf of the Hungarian Biophysical Society and the Hungarian Academy of Sciences Biophysics Scientific Committee, and I would like to ask, my name is Georg Panyi, I'm from Debrecen, University of Debrecen, Department of Biophysics and Cell Biology, and I would like to ask my colleague, Professor Kellermayer from Semmelweis, to greet the audience with a few words.
Thank you very much. Ladies and gentlemen, dear lecturers, it is truly my honor to join Professor Panyi in welcoming all of you and praising the organization of this symposium, and thanking all of you for joining us here. It is indeed a celebration of science throughout this month in Hungary, and celebration of biophysics, and within that the celebration of von Békésy, who was a main contributor to our understanding of how hearing works. And today we'll be celebrating it together with, we are giving a due tribute to his accomplishments by inviting the most notable contributors to this field. So with that, I want to give back the word to my colleague, Professor Panyi, and I look very much forward to this exciting symposium.
Okay, and as a first speaker, I would like to call Professor Péter Dallos, who is going to give an introduction, and he's currently in the United States, and he's joining us on Zoom, and the Zoom broadcast is supposed to be starting right now. Thank you very much for accepting our invitation.
Kedves barátaim and kollégák, dear friends and colleagues, I met Békésy once when he came to give a talk at my institution in Chicago. I took him out for dinner, and we had an extremely spirited discussion about art. He came to Chicago because the only reason he was willing to, because he wanted to visit the Oriental Institute, where he loved to see the Middle Eastern and Far Eastern art. Our discussion was that he hated contemporary art, which I collected. So as you can imagine, we had a very good time talking about. He also finished probably the biggest rare steak I have ever seen in my life, which can only be had in Chicago restaurants, and then he proceeded to drink me under the table. You might want to ask, why to have a symposium to honor someone 50 years after his death? It's very simple. If you look at, say, Web of Science or Google Scholar, to find how cited his work still is, you will find that yearly he has more citations now than most scientists have in a lifetime. That's a good reason. Just as with other branches of science, life sciences in the past 50 years, auditory neuroscience has made extraordinary strides. While the post-war period research was conducted in a very few laboratories with highly influential players such as Békésy, Davis, Weaver, Nelson Kiang, and György Rose. The past 50 years is characterized by an ever-expanding cadre of first-rate researchers who collectively produced our current understanding of the auditory system, incomplete as it may still be. So I will just spend a very few minutes in giving you a few highlights of these last 50 years. We now know that Békésy's singular achievement of discovering the traveling wave and thereby establishing mechanical frequency analysis of the basilar membrane in the mammalian cochlea applies only to dead ears and to very high sound levels. It's very interesting to remember a meeting in 1969 in Holland where Brian Johnstone first presented evidence that, in fact, basilar membrane tuning was sharp in the live animal, unlike Békésy's, similar to neural tuning, as established by the work of Bob Galambos and Nelson Kiang. No one believed Brian. Such was Békésy's influence. Virtually the entire meeting was spent by telling Brian how he was wrong. Not until the seminal work of Bill Roddy, who showed that basilar membrane tuning was nonlinear and physiologically vulnerable, had the field abandoned the notion that extraordinary frequency resolution of the ear is somehow produced by some mythical second filter interposed between Békésy's broadly tuned basilar membrane motion and sharply tuned auditory nerve fibers. When David Kemp demonstrated autoacoustic emissions, validating Tommy Gould's 1948 suggestions that mechanical amplification in the cochlea became established. The big question remained, how was that established? When I came into this business in the mid-1960s, Hal Davis's proposal ruled, namely, that in some analogy with retinal cones and rods, inner hair cells processed high-level sounds while outer hair cells handled low-level ones. With Heinrich Spundlin's demonstration of radically different afferent and efferent innervation patterns to the two types of hair cell, a new realization dawned. Inner hair cells communicated auditory information to the central nervous system via their rich afferent innervation, while outer hair cell did what? It is quite amusing to read today from today's perspective, the multitudes of imaginative theories promulgated in the late 1960s, early 1970s, all which were designed to answer this question: what do outer hair cells do? When Alan Ryan and I showed in 1975 that outer hair cells were in fact amplifiers producing some 50 decibel gain, and when Dave Harris and I showed in 1978 that in the absence of outer hair cells, auditory neurons had raised thresholds and poor tuning, that's when the multitudes of theories were overtaken by experimental results. Outer hair cells function as the cochlear amplifier in mammals. The next pivotal question of how do they amplify was partially solved by Bill Brownell, who demonstrated electromotility of outer hair cells in 1985. This voltage to cell length change of outer hair cells naturally resulted in a feedback loop in which Békésy's traveling wave is amplified by mechanical feedback from motile outer hair cells. The next fundamental matter to be resolved was how are outer hair cells capable of changing their length to provide this feedback. This was clarified by my laboratory in the discovery in 2000 of the motor protein Prestin, a piezoelectric direct voltage to displacement converter protein. The role of Prestin as the basis of amplification was proved by Charlie Lieberman's group, showing that Prestin, knockout mice, suffered some 50 dB hearing loss, just as our 1975 outer hair cell deficient animals. Finally, very recent work on the structural description of the molecular structure of Prestin concluded a 20-year quest. You will hear more about this from Eduardo. Parallel with all this, there was tremendous progress that had been made on elucidating the properties of hair cell mechanoelectric transduction channels, starting with the seminal work of Robert Fattyplace, Jim Hudspeth, and David Corey. Largely in non-mammal vertebrates, the fundamental properties of these ion channels were established. Most importantly, they also showed that displacement induced deflection of the stereocilia, which are the input organelles of these cells that contain the channels, is reciprocal. The cilia are able to produce force in response to their deflection and thereby amplify sounds. The stereociliary feedback has been shown to produce amplification in many non-mammalian vertebrates, just as Prestin does in mammals. Jim, David, Dolores, and Yuli will talk about these remarkable ion channels, including recent results in identifying their molecular components. Enjoy. Thank you.
Thank you very much, Professor Dallos. And I would like to ask if anybody has questions to Professor Dallos from the audience. And in the meantime, I would like to ask Mariann Békésy to prepare with for her presentation. Any questions to Professor Dallos? I do not see in the audience. So at this time, I would like to take the opportunity to thank all the speakers for their participation. And first, to Professor Péter Dallos. And I would like to present a certificate of acknowledgement for his contribution, which hopefully can be seen on the screen as well. And most importantly, we have prepared, with the help of Kis József Géza, a Békésy medal, which will hopefully make sure that this seminar will be remembered for a long period of time. So thank you very much for Professor Dallos, and definitely we will make sure you will receive this this diploma as well as the medal in a short period of time. Thank you very much again.
Thank you.
And my privilege also to call on the podium Mariann Békésy, who is the niece of Georg von Békésy, and she's going to talk about the heritage of Georg von Békésy. So, Mariann, please go ahead.
Dear Chairman and audience, thank you very much, Professor Kellermayer and Panyi, for the organization of the conference and for the invitation to be part of the commemoration. Given that for Békésy, art was nearly as important as science. So I decided to take some thoughts from his lecture, Art in Science and Science in Art. My goal is to give you a flavor of his joy and thoughts. Art helped him getting through the difficulties, and art knowledge created a value in his art collection, what he left to the Nobel Foundation. At the beginning of the Nobel lecture, he stated, "There is one thing I am always ready to do, and that is to look at beautiful things. I can look at an art object for hours, and I am convinced that I owe a large part of my education of the museums of many countries." But how does art help a researcher to achieve success? At a young age, he interested in the ways to become successful. Observing the history of successful artists, he recognized that success depends on quality and quantity. Success equals quantity times quality. Either is zero, the result is zero. Comparing success in these two areas, quality is more important even in art, where the value is subjective. In the objective science, quality is even more important. As a student, he was concerned with why it's so difficult to imagine something new. It is difficult to recognize these in books of chemistry and physics, but it is easy to see them in the history of art. Between the 12th and the 15th century, it was a custom in Europe to use fantastic animals for decoration. If you compare the figures, you will see how limited the fantasy was. The figures are nothing else than combination of other parts of other figures. But when your imagination is so limited, how is it possible to produce new discoveries in science? It was the study of Leonardo da Vinci that gave him the answer. If you compare the drawing of Lower with the drawing of the storm, you will have the impression that Da Vinci was able to cover a velocity range that no other artist had been able to equal. Why? Because Da Vinci didn't try to outdo nature with his fantasy, but he tried to learn from the nature. At the university, he was interested in surface chemistry, which was thought to be the key to life at that time. He collected and had all available books on the topic in the library and began to study them and compare them. Soon was able to determine which was better and why. Using this approach, he read 4 or 5 times as much as the other students. He deducted that the only way to understand an object is to view from several perspectives. So, to perform quality work, the best way is to compare, and that was he wanted to achieve. Soon thereafter, an art dealer validated his approach. Békésy asked him how he can distinguish the original from the replication. The art dealer explained that the accurate observation and comparison is the only way to distinguish one from the other. Following this, Békésy began to compare and compare again. The first The first comparison was an amusing story. A well-known art dealer recommended for purchase a unique, beautiful Mexican jaguar stone sculpture. He was not yet proficient in Mexican art, but he was affected by the face of the jaguar. The art dealer allowed him to take. The statue stood on the desk about 3 months, and he always enjoyed it. Then on a peaceful and quiet Sunday morning, when he relaxed looked at the jaguar, he had the feeling that the jaguar is laughing at him. He got excited and turned at the statue, but it seemed always laughing. The jaguar statues are ritual objects. The most have a serious face. A smiling jaguar cannot be an original one. He took back the jaguar, saying that it is smiling, and the dealer agreed. And he told that he will take back the sculpture if it turned out that it is fake. And it would be a good lesson for both. The statue weight was 15 kilograms, and he took it from Boston to New York to show to the best expert. When he entered, he had to smile, because just like another specimen saw him there. Probably a Spanish aristocrat made 2 sculptures for his gateways at that time after Columbus has arrived, so the jaguar couldn't be a pre-Columbian one. According to the lesson of history, elegance is a result of hard work, both in art and science. Not the same as beauty. Beauty is, for example, for a young lady, is a hereditary thing. But elegance, a kimono or a graceful sculpture, a beautiful dance, it is a result of hard work. After the First World War, and in a very bad economic and political situation, he decided to work for nothing at beginner. This approach worked fine because the only laboratory in Hungary which was well equipped in 1920 was the Post-Experimental Institute. The important telephone lines went through Hungary. There were constant complaints that Hungary didn't keep its lines in order. The test method used took more than 20 minutes several times a day, and everything stopped. He wanted to find a method could complete a test in a second. When a musician tests his violin by plucking the string, he can tune immediately. So he thought that plucking the telephone line should be possible to obtain instantly the same data as that are obtained in the cumbersome 20-minute test. He transmitted a click through the line by switching a small DC voltage and listened and observed the returning signal. There was a DC difference, and shortly he was able to tell the site of the disturbance. The next question was, what will be the future development of telephone communication? The improvement of the long-distance cable, or the telephone, or the microphone, or the switchboard? Where to invest the money? His opinion was that the least development would produce the largest improvement of the whole transmission system. He started investigating the quality of the different parts. It turned out, in a very simple measurement, that the microphone is the poorest. They should finance the research which may improve it. The test was simply plug the telephone membrane and determine the resonance frequency and the damping of the membrane. By comparing these two numbers with the ear drum, the membrane turned out to be very, very poor. So he started to investigate the ear, so he turned back to the nature to learn from her. The cochlear is a small organ located in a very hard part of the skull in the os petrosum. His first trial was in the Anatomic Institute, but the anatomists couldn't dissect the cochlear without hurting it. Not by chance, he wanted to try himself. He finished the high school in Bern earlier as he could start the university. He used the remaining time to learn Swiss precision technical skills at Hassler Company. So he could prepare the cochlear, and he accomplished the saying in the family, fortune will only find those who are sufficiently prepared for it. When he was able to see the cochlear, the beauty he experienced impressed upon him to stay with the ear research. And when the success came, the title of his Nobel lecture articulates the driving force of his whole life and work, the pleasure of work, research, art, and science. Thank you very much.
Thank you very much for the presentation. It is a real pleasure to hear from the first person that had the family heritage, and also thank you very much for a very, very interesting summary. And at this moment, I would like to thank you also for the participation and congratulations. This is the medal
Thank you so much.
for you. And I would like to present the diploma. Also, can you come a bit closer so that the cameraman can also see? And well, thank you very
so
much.
much.
It
Thank
was
you
a real
so much.
pleasure. Thank you.
Thank you.
Okay, and now moving on, as Péter Dallos has proposed, we are going to be hearing from the first hand about the mechanism, how the cochlear amplifier works. And this was recently discovered by my friend Eduardo Perozo, who is at the University of Chicago, and he's going to talk about Prestin conformational dynamics and the molecular basis of cochlear amplifier. And we are very happy to have you on the meeting, and please go ahead, Eduardo.
Hello, it's a true pleasure to be here. In fact, I should have been there otherwise for recent COVID infection, but it is an honor to be a participant in this symposium. What I would, as you know, Péter Dallos introduced remarkably well the topic. I'd like to talk to you about cochlear amplification. But first, you know, I'd like to honor the people who actually did the work. This is a remarkable collaborative effort in my lab, driven mostly by a very talented postdoc, Navid Babi, who's here and at the microscope. And it is also more recent work in collaboration with the Sussex lab here at the University of Chicago, which I'm going to be talking some unpublished work on the hydrogen deuterium exchange with mass spec as a way to start understanding the basic principles of the voltage-dependent part of Prestin. You know, many of the talks today will actually deal with this basic introduction. Excuse me. You know, hearing, as the remarkable and pioneering work of von Békésy showed, is sort of a center in the inner ear, of course, and the transduction occurs in the cochlea, there are two basic fundamental mechanisms. One that will be explained and discussed extensively in the next few talks, is the actual transduction of how we transduce pressure waves into electrical activity. I will not talk about that. The second one, which Peter nicely introduced, is the question of amplification. If we don't amplify, and this is all work, as Peter mentioned, all of the work from Békésy was done in what is called the dead cochlea. There is a known experimental amplification that occurs about 50 dB, mostly at high frequencies. And I'm going to be talking about the molecular basis of this process. This, as you know, if you sit on the traveling wave, there conformational transitions that lead to motion of the basilar membrane, and the role of the outer hair cells is critical. And this, of course, produces a kind of, and under debate whether it's cycle by cycle or there's an additional issues of intrinsic filtering, which I will deal at the very end of the talk, as you'll see. These motions produce a local amplification that drive the gains at higher frequencies, as mentioned. Also, as Peter mentioned, you know, one of the key, there's a couple of key results that have been shown. One is the discovery of electromotility and the role of the outer hair cell in all this amplification process. In short, outer hair cells behave as short contractile systems that are voltage dependent. And this voltage dependent relates to the motion, as I said, at high frequencies, where depolarizations lead to a shrinkage of the outer hair cell, whereas hyperpolarization extends the length of the cell. This has been characterized to a high degree, at least at the cellular level. And I'd like to show you an example that was recorded many years ago by Jonathan Ashford's lab. He's actually gotten a lot of mileage out of this beautiful system where you can voltage clamp an outer hair cell, but instead of showing a square wave, you place an actual song into it, an MP3. And at the end of the day, by controlling the voltage, you make the cell actually dance. All right, enough of that. You can measure this, both electromotility and the voltage sensitivity part of it, by two measurements. One, optical measurements allow you to describe the electromotility at nanometer level resolution. And then there's a voltage dependence to it. Let's see, can I get, and then we can measure that, the component of the voltage dependent of the electromotility by measuring nonlinear capacitances, which essentially are the electrical signature of the conformational changes of the molecular motor associated with electromotility, here shown in the bell shape curve. For somebody like me that comes from the voltage-dependent ion channel field, you know, you can explain this very easily. This is the first derivative of the charge movement translating across the membrane. So the molecular underpinnings of this process, of course, were clarified at the beginning of the 2000s by the pioneering work from Péter Dallos, which is covered that this voltage-dependent motor in outer hair cells is no other than Prestin. Prestin, in fact, belongs to, is not an ion channel. If anything, it may be considered as a broken transporter because it belongs to the major solid facilitator transporter family, the SLC. In particular, Prestin is referred technically as SLC 26A5, which is a small family of organic anion transporters that are driven by chloride, typically. The interesting thing about the discovery and characterization, molecular characterization of Prestin, is that these type of transporters tend to belong to a family that whose mechanism is associated to what is called the elevator transport mechanism, where there is a moving part that leads to three major conformations, where a binding site for the transporting solute is actually characterized by access to the outer extracellular solution, intracellular solution or inner access, and there is a third component, the conformation, that is occluded in the middle. So a few years ago, we were very interested in this process at the molecular level because well, there was no indication of what the molecular mechanism of something like a transporter will lead to amplification in the inner ear. Naively, we thought that by having systems that respond at very high frequencies, like bats or dolphins and whales, will provide a robust target for biochemical characterization, expression, purification, and ultimately the structural determination. This turned out not to be the case. I mean, we can actually purify Prestin from a number of different sources. Other labs have done it, as I'll describe shortly. But we focus on the whale Prestin or dolphin Prestin. It's very similar there. It has to be toothed whales. We're able to purify, characterize. We can take this open reading frame, express it in a heterologous system like hex cells, and not only obtain evidence for nonlinear capacitance in the frequency domain, but also in time domain. But most importantly, we're able to reconstitute the electromotility. As you can see here, as we apply different voltage clamp pulses, you can start seeing the hex cells exhibit electromotility only by adding Prestin. Also importantly, this electromotility is inhibited by the best known specific inhibitor of Prestin activity, which happens to be salicylate. All of this electromotility and nonlinear capacitance in this cellularly reconstituted electromotility in X cells can be inhibited by salicylate. So I will save you all the gory detail of how we got the structure. The structure was obtained a couple of years ago, and as predicted from its evolutionary relationship, it looks like a SLC type transporter. This type of transporter, as I said, has three different components. There is a cytoplasmic domain for which most of the somatic mutations that affect Prestin and electromotility in outer hair cells can be found. But the active site of all of the electromotility origins by Prestin are, without a doubt, characterized or located in the transmembrane segments, of which there's two components. In the field, as in comparison with most transporters for which we know the structure, there's two parts: the gate domain, which I would more functionally refer to as the scaffold domain, and the core domain. The core domain is the one that produces the transport. This is a moving part. And in a system like this, we can make a caricature. The scaffold is the reference point to the movement of this elevator domain, for which there is an anion binding site. In the early 2000s, Péter Dallos and Bernd Fackler's lab demonstrated the fundamental importance of chloride, or in fact, many different small anions in the function of Prestin. And there are a number of properties of the Prestin structure that provide clear information on where the anion binding is and how potentially it binds and produces conformational transitions, which is what I'm going to be talking to you about. So if we take a monomer out of the dimeric structure of Prestin, you see that this is a domain swap a dimer structure where one subunit intertwines the second one in the between transmembrane and cytoplasmic domain. If we take only the monomer, we see that there are clear indications of where the anion binds. This comes from experimental data on transport, mutagenesis, and obviously the structure. There are 3 helices associated with it, in more or less inert helix called the TM1, which is at the far periphery of the structure. Internally, there's 2 short helices that produce what is called a dielectric binding site. And this dielectric binding site contains not only aromatic probably pi anion interactions, but also a fixed charge of 399. And as we'll see in a few minutes, this is of fundamental importance to the defining not only voltage dependence, but anion binding and the dichotomy between those two. Then we can clearly place the chloride bound in the middle of this dielectric gap. This dielectric gap, of course, allows for a partial charge from the microdipole of these 2 short helices to stabilize electrostatically chloride, in addition to the electrostatic stabilization by 399, R399. This is very referential to what is known in classical chloride selective anion channels, where a similar dielectric gap contributes to the binding of chloride. So for the rest of the talk, I'm going to be dealing with 3 major questions, maybe 4 if I have time at the end. One is I would like to define the set of conformational changes in 3 dimensions at the molecular level of what Prestin's conformational landscape may be. Then using this information of what Prestin motions are, and there are motions associated with putatively voltage transitions, how does that contribute to the conversion of membrane potential into mechanical movement and vice versa? Then I'm going to end with the insights on what the nature of this dual voltage sensor in Prestin is, and then to finally understanding cochlear amplification, which we don't fully yet. I'd like to make a couple of comments on going from these atomic coordinates to the mesoscale behavior at the cellular level, which I think is where the key questions remain in the future. In order to get to this conformational landscape, of course, we have to address what the major challenges are. In any voltage-dependent system, and in Prestin, as you'll see, we have 2 components: the electromotility part of it, the motor part of it, and the voltage sensing part of it. So for any voltage-dependent system, we need to modify the landscape by changing the nature of the electric field across a putative sensor. This is the case for most voltage-dependent sensors in ion channels, and you go from, say, a down conformation to an up or activated conformation. If we were to do this with structural techniques, we could either do it with crystallography, which nobody's been able to crystallize Prestin yet, but it requires the generation or the imposition of very large electric fields across the crystal, and that is technically feasible but very difficult. Equally difficult is the uniform application of a compartmentalized system, like say a liposome reconstituted Prestin, in which not only we have to apply the field homogeneously and simultaneously, but we need to account for the orientation of Prestin in the electric field. Instead, we've taken advantage of a very clever trick. In the early 2000s, mid-2000s, it was discovered that the voltage dependence of Prestin is very much dependent on the type of anion that is not only physiologically present in outer hair cells, like chloride, but any other anion that can compete with chloride to that binding site that I mentioned earlier. And in fact, in chloride, we see the standard behavior of the nonlinear capacitance as we substitute that binding site or compete it with either phosphate or sulfate. We see significant shifts towards the more depolarized potentials. That means that at 0 millivolts, where all of this cryo-electron microscopy structures are obtained, we can actually sample the two sides of the distribution of nonlinear capacitance, either in the once Prestin is active, at or close to its peak, or past its peak. And this is exactly what we've done by characterizing dolphin Prestin nonlinear capacitances in different anions, as was done earlier in the 2000s. And we see that in chloride, we do find sampling at 0 millivolts past its peak, past its midpoint of activation, whereas exchanging chloride by sulfate, we get to sample mostly or close to the peak of the nonlinear capacitor. This has two key predictions. One is that in chloride, the conformational heterogeneity obtained by cryo-electron microscopy will be limited mostly to the activated form of Prestin, or what we call the up form. And in sulfate, we are bound to see a much more heterogeneous distribution. If you look at any sort of basic modeling based on Boltzmann distributions of conformational transitions, the prediction for Boltzmann is that the maximum entropy of the system will be localized at its peak. And this is exactly what we see. In chloride, we see one conformation mostly, which from now on we're going to call the elevator up, or compact conformation. Whereas in sulfate, we see a distribution of multiple conformers. We see two expanded or down conformation and an intermediate in between conformation. And I will sort of elaborate on that in a second. In addition to that, we can expand the conformational landscape by inhibiting Prestin in the presence of salicylate. In salicylate and or either chloride or sulfate, we not only see the bound inhibitor right in the dielectric gap, as I mentioned before, which means that it competes directly with chloride for binding. It expands the nature of the dielectric gap, which then changes the local electric field across the binding site. And so under these conditions, we see, and this will be expanded shortly, that an adjacent helix, TM6, next to the binding site, it will be probably at this direction. We see a hinge bending motion of TM6, which has fundamental consequences into what we think is the whole behavior and expansion of the conformation of Prestin. So let me summarize what I said so far by doing simple a linear interpolation between the different conformations that I've mentioned. One, if we fix the reference point at the elevator domain, the movement, you see that the movement occurs not only there is a component in the z direction, but there's an ample change in the plane of the bilayer, and I'll go into detail in a second. But if you focus the reference point at the binding site for chloride, you see an accordion-like behavior of the dielectric gap. And this may be critical to understand how binding and unbinding of chloride couples to the overall conformational changes of the elevator. All right, this is again a summary now in cartoon form, where I summarize the situation of the structures in chloride, the structures in sulfate, and the structures in sulfate and salicylate. In each case, we can unveil a different conformation. So I want to highlight the fact that as we publish this, and about the time we published this, Eric Guo group at the Volume Institute showed very similar structures of these two conformations. They didn't see evidence for the electromotility elbow, which I'm going to show in the next two slides. Santos Sachi's group, which has been also a pioneer and contributed in this field, has shown the chloride structure. And most recently, the Nueki group in Japan has shown, again, the stable up or compacted conformation by engineering Prestin with about 40 different mutants, but essentially it's the same conformation as we show here. Now, based on what I've just showed you in terms of the pure structure, the main challenge is how to interpret this in terms of conformational transitions and how these changes in voltage lead to expansion in the plane of the bilayer. So one of the things that we do, obviously, is to look into the cross-sectional area changes between Prestin. As an example, for comparison purposes, we use a clear transporter. It's a mouse oxalate transporter, SLC26A9, which is the closest structure we know from the structure of Prestin. In this case, we see changes only in the Z direction in the outer leaflet of the membrane, whereas very little changes occur in the inner, outer leaflet here. Very little changes occur in the inner leaflet of the bilayer. And this comes as a consequence, of, as I said, of the multiple components in 3 dimensions of these conformational transitions due by the elevator motion. In addition to that, so we see that when we model these changes in the presence of a lipid bilayer using molecular dynamic simulations, we see a clear thinning of the bilayer right next to the TM6 in the inner leaflet of the bilayer. And we think that this is the key actuator, mechanical actuator for Prestin in the outer hair cell. Now, experimentally, we can see these are structures are obtained in detergent, but we see major changes in the detergent architecture in the micelle surrounding Prestin, depending on whether we stabilize its compact or its expanded conformation. And as you can see here, there's 2 components. There is a saddle point here in chloride. It flattens in sulfate, but in addition to that, it produces a large cavity driven around the helix TM6. Not only that, you can see it from the top projection that the micelle compresses and expands in 2 different places. This, of course, will require additional experimental evidence in either the cellular context or in large enough nanodiscs that doesn't limit the conformational flexibility, as is the case now. Let me go back to this unusual observation of the electromotility elbow. TM6 produces this very large hinge bending motion, and this motion is exerted also by the fact that the end terminal end of the TM6 has a large proportion of charge groups. This is, we think, the origin of this gap in the micelle that produces narrowing, shortening of the bilayer of the micelle at the end of Prestin. When we look at this, we like to make an evolutionary observation in that Prestin and electromotility, as properly known, is a recent phenomenon from the evolutionary point of view. It's only present in mammals. And when we look at the composition of TM6, it has a very large number of glycines. In fact, it's a total of 5 glycines in mammals, and only 2 in non-mammals, from chicken to, say, turtles or even fish. So we focus our attention in looking at the mechanistic contributions of this TM6 and this electromotility elbow to expansion in Prestin and the function of electromotility. And in fact, mutating those mammal-only glycines, and particularly this glycine duet here, 275 and 276, it turns out that mutating any of these to what is found in amphibians and fish completely abolishes, or at least shifts, the voltage dependent to points where, physiologically speaking, there is no nonlinear capacitance and no electromotility. And we've been playing around with mutations around these glycines to show clearly that this electromotility has fundamental properties contributing to not only nonlinear capacitance, but electromotility expansion and contraction. So having said that, then the question remains, what's the nature of the voltage sensor? And here's a tricky question from a mechanistic aspect, a mechanistic point of view, because voltage dependence in Prestin, it has a dual component. On one part, there are a number of charges distributed along Prestin and before the structure. And this work from, say, Santosacchi's lab from about 13 years ago focuses on every single charge that is not 399. And when we map those charges, we see that they're very subtle effects from the point of view of voltage sensing itself. The changes in the estimated charge movement is in the fractions of a charge. And that's not surprising, since most of these charges are located mostly outside the electric field. The only one that seems to matter is the location of the 399, as I said, that contributes to the binding of chloride. If we compare two systems, Prestin, which has electromotility and nonlinear capacitance, versus the transporter that has neither, we see clearly that 399 is placed in a position to influence not only the local dielectric field, but the binding of the chloride itself. And so this duality between 399, which we can mutate and completely eliminate, or at least shift the voltage dependence to non-physiological ranges, and also this dependence to chloride, was sort of a conundrum before. So we decided to use hydrodeuteric exchange using mass spec to look into this problem. In short, we can look at the conformational changes and the local dynamics of any protein by doing deuterium exchange, where we quench a different pH, proteolyze the peptide, and then use mass spec to analyze the fraction of those peptides for which there is the hydrogens have been exchanged to deuterium at different times. So for the transporter, and we focus on peptides that sample the dielectric gap here, which will be peptides A, B, and C, color-coded here. We see that not only is the dielectric gap and the chloride binding site quite robust, it also shows very stable dynamics. As in comparison with Prestin, this produces a fairly narrow and destabilizing effects as we decrease the chloride concentration. As you can see, this is summarized here. This sigma expansion protocol, which gives you an estimate of how stable a particular region of the dielectric gap is, we see that in the transporter, we see a cooperative unfolding, which means that in the absence of chloride, it's stable by its binding itself, and as soon as it reaches a threshold point of concentration, it completely unwinds, all cooperatively. In contrast, when we look at the behavior of chloride dependence of the dielectric gap in both TM3 and TM10, we see that there is what is called fraying, which means that the stability of the system depends almost linearly with the chloride concentration. This dependence suggests to us a potential mechanism by which the location of the 399, the guanidinium moiety of the 399, is closely coupled to the presence of chloride. In the presence of chloride, 399 is located so that it feels the electric field. In the absence of chloride, this fraying potentially drives the guanidinium outside of the electric field, leading to a loss of voltage dependence. So this needs to be confirmed with structure, which we are currently pursuing vigorously, but this provides a very clear explanation, in my mind, to this duality between chloride binding and the presence of a fixed charge in the voltage sensing part of Prestin. So this is summarized here. We can either have 399 in the electric field in the presence of chloride. The chloride is also within the field, although chloride itself and the anion does not contribute to the gating charge and nonlinear capacitance. It is an indirect regulator of the conformational flexibility of this 399, which appears to be the true sensor itself. But in addition to that, as I mentioned before, during electromotility, there is a shortening, a narrowing of the length of the bilayer, which will intrinsically affect the capacitance. So it's a very complex, multi-dimensional effect that produces voltage dependency. Finally, I want to make a couple of comments instead of understanding what I've shown you at the molecular level with what happens at the cellular level. And this is where the major gaps and future directions are, I think, in the field. A back of the envelope calculation of the types of motions that we've shown tend to support this idea of the area expansion model. And the area expansion model, as suggested about 10, 20 years ago, depends on the density of prestin in the lateral membrane of the outer hair cells. This is a work from Hay et al., where you can see clearly from wild-type outer hair cells, the density of particles dramatically changes between wild-type and prestin knockouts. And if you take that as an experimental value on average, you can estimate that there are about between 5,000 to 10,000 prestin copies per square micron. This is not a two-dimensional crystal, but it clearly approaches densities that can be seen, say, for the sodium channel in the nodal or VA. This is a very intriguing problem in terms of cell biology. However, we can take that information and do a back-of-the-envelope calculation using known parameters on experimental parameters on outer hair cell. And we see that if we just input this into a very simple model where we maximize the orientation and the orientation of prestin relative to each other is fixed, we can actually calculate about a 2.6 micrometer motion based on the expansion of the conformations in prestin that I showed you earlier. Recent work by Emad Tarkosh's lab, this is all theoretically, suggests that this orientation of prestin could be maximized at about 45 or 135 degree orientation relative to each other. And then there is cross-talk through the bilayer, which we think is the explanation for this thinning of the bilayer that I mentioned before. Now, is this enough to understand electromotility and more importantly, the cochlear amplifier? Recently, there's been a heated debate of whether or not outer hair cells have what is called the speed paradox, where if you look at the electrical properties of the outer hair cells, there is an intrinsic built-in filter that sort of has a corner frequency around 5 to 6 kilohertz. If you assume this, what is called the cycle-by-cycle hypothesis, where every time the transduction occurs in the stereocilia, it changes the transmembrane potential, which then drives prestin conformation on at each frequency point. This frequency filtering that occurs electrically should not be explaining on a cycle-by-cycle basis. There are arguments pro and against currently in the literature, and I think that this is one of the most exciting aspects of understanding cellular electromotility on the basis of conformational transitions. So just to conclude, I'm going to show you a little movie that summarizes what I mentioned before. Prestin functions as a true piezoelectric device where charge movement is associated to the expansion of a dimer in the membrane that occurs at very high densities in our hair cells. And this expansion can be quantified up to 12% of the cross-sectional area, mostly in the inner leaflet of the bilayer. Translating these motion to at the molecular level to the cellular level remains one of the key questions in the field. And with that, I thank you very much for your attention, and I'm open to questions.
Thank you very much, Eduardo. It was a very challenging task that you performed, and I wish you the best for continuing this work and quick recovery. And we are really sorry that you couldn't make it here, because your presence would have been really a highlight of this symposium here. I would like to ask the audience if there is any question, a very short one to Eduardo, and then at the same time, anybody, any questions to the speaker? If not, then we would like to thank again Eduardo, and we will definitely make sure that you're going to be receiving the diploma as well as the Békésy medal, okay, as soon as possible. And with that one said, we are moving forward, and the next speaker.
Thank you very much.
Yeah, thank you very much, Eduardo, again. Thank you. With this, we would like to move on to Uli Mueller, who is from the Johns Hopkins University, and he's going to talk about the another ion channel, which is the Mechanotransduction channel complex of cochlear hair cells. So I would like to ask Uli to Ulrich Mueller to start his talk.
György, thank you very much. Can you hear me?
We can hear you very well. Thank you.
Thank you very much. György and your team, I first of all would really like you very much for organizing this symposium in honor really of a founding father of our field, Georg von Békésy. It's a great honor to be here, unfortunately not in person, by Zoom. I hope we can rectify that at some point. I think we just from Eduardo, we've got a wonderful example how the field has moved from physiological and biophysical principles to really an atomic structure understanding of what is the underlying molecular basis of this amplification process in mammals. What I want some resin? Okay. What I want to talk today about is an equally, I think, fantastic journey which has led to an understanding of the core components of the molecular machinery that underlies the conversion of the mechanical signals in the cochlear into electrical signals. And as we, of course, all know here in the audience, I'm sure, is that the hair cells in mammals are embedded in this cochlear end organ here, the organ of corti, and these hair cells have this absolute beautiful shape, this long cell body crowned by the stereocilia. And these stereocilia shown here in mammals in higher resolution have really this very fantastic shape. They have this amazing organization where we have three stereocilia of graded height that are connected at the upper end by these little tip-link filaments, which were discovered by Jim Pickles in 1984. Now, what's really foundational work in the field carried out by Jim Hudspeth, David Corey, Robert Fettyplace, and their co-workers has revealed how this mechanically organelle actually transforms a mechanical into an electric signal. And what these studies have revealed that this organ has that this polarity in the stereocilia is very important for function, where deflections in the directions of the longest stereocilia lead to the opening of ion channels within the stereocilia bundle. These ion channels are non-selective cation channels, and they had really eluded discovery for decades, as had the identification of the tip-link filament. What are the molecular components had really remained a mystery. And one of the big problems had been that hair cells are really encrusted into this organ, which is shielded by the temporal bone, the hardest bone in our body, really, and they're few in numbers. So biochemical approaches had been very difficult at the time, if not impossible, to identify the component. So a variety of laboratories, including ours, have explored the genetics of hearing loss to identify components of the mechanotransduction machinery. And that is based on the observation that deafness is the most common form of sensory impairment in humans, and it is estimated roughly mutations in 500 genes can lead to deafness. By now, we have positionally cloned about 100 different genes that are linked to the disease. And what is very truly remarkable, that for about 70 of these genes, we have mouse mutants. And the mouse mutants are very, very informative because the study of the mice and the humans has shown that there's nearly 100% conservation in the genetic landscape of deafness between mice and humans. So whenever you have a gene that causes deafness in humans and you make a mouse model, the mouse will suffer from deafness. And whenever you identify a mouse mutation which leads to deafness in a new gene, you usually can find a human family which has a mutation in that gene. And the study of these genes really has driven the discovery in the field related to mechanoelectric transduction and to other phenomena. And what my laboratory has actually done, we've taken roughly 70 of these mouse lines, and we have studied the mechanotransduction process. And the way this is done is really in an organ prep, where you take a hair cell, you patch on the cell, and you stimulate the hair bundle. And then you can record from the cells these currents. When you stimulate the bundle very strongly, you get a very maximal current from the hair bundle, which then adapts. If you have a mutant mouse line, here's one example shown, that affects transduction, the currents may be severely impaired. This here are single recordings, but you can, of course, statistically analyze this for many hair cells, and you see a current displacement plot, which is typical for wild-type hair cells, and you have one mutant, which shows severe reduction in the response to mechanical stimulus. And using this approach, we have by now identified 17 genes which are linked to mechanotransduction. As I will show you, we haven't published all of them yet, but many of them encode components of the transduction machinery, and some regulate the assembly of the machinery. Now, the first study, genes that were really informative were these genes linked to Usher syndrome. Studies which were spearheaded by two graduate students in my laboratory, Jan Siemens, Piotr Kaczmirczak, and the work from also other laboratories, most prominent from Zubair Ahmed's lab, identified the core components of this mechanotransducting important elements, a tip link, showing that these ones are formed by Cartesian 23 dimers that interact with Cartesian 15 dimers to form this beautiful asymmetric filament. So suggesting already that there may be functional asymmetry in the tip link. Work from our lab, Christine Petit's lab, and Bishara Kashar's lab demonstrated that there's a protein complex at the upper end of the tip link which likely adjusts tension in this complex. Mutations in any of the genes shown here leads to this disease Usher syndrome. So one common component then of the disease is really a failure of this mechanotransduction machinery. Now, what the original studies did not identify is the components of the channel itself. However, a very nice study from the Ritchie and Fettyplace laboratory provided evidence that the ion channel is localized at the lower end of tip lengths. And they took advantage of the observation that calcium ions can penetrate through the channel. So they observed where calcium actually enters stereocilia and showed it's really close to the lower end of the tip length. And that leads then really to this model, extends this, that there's this wonderful molecular asymmetry in the complex with two functionally distinct ends at the tip length. Now, the rest of my talk, I will describe work on the molecular identification of the ion channel complex, and also some new studies which are not published yet on the gating mechanism. Now, one assumption then from this, as highlighted from the previous studies, is that the ion channel should be very close to the lower end of tip length where protocartian 15 is localized. And in 2013 and 2014, two molecules were identified that might fit the bill. My laboratory and a postdoc, Bo Zhao, who is now at the University of Indiana, spearheaded this work. He showed that this little protein called TMIE, which is linked to deafness in humans as well, is linked near the lower end of tip lengths. And Jeff Halt's laboratory, in collaboration with Andy Griffith, showed that this molecule TMC1 and its close homologue TMC2 are localized at near the lower end of tip lengths. When you when you make mutations in TMC1/2, you have to mutate them both, or mutations in TMIE, you abolish mechanotransduction in hair cells. So you see here's a wild-type current that you get from a wild-type mouse, and down here's the traces you would see in the mutant. The currents are completely abolished. Now, a breakthrough in the field then came when it was discovered that TMC1 has a sequence homology to a protein called TMEM16A, an ion channel whose cryo-EM structure was solved several years ago. The laboratory of David Corey and Canton Swartz, they developed molecular models of TMC based on the TMEM16 structure, which predicted that TMC1 would be a dimer on each of the molecules having 10 transmembrane domains. Jeff Holt's laboratory tested this model because this model also predicted that here on the outside of this molecule would be an ion channel pore shown here in the space-filling diagram. So we have here the transmembrane domain, this TME, and here the pore domain. So what is peculiar about this pore domain, predicted pore domain, would actually be open to the lipid environment when you imagine this here being embedded in the membrane. So what Jeff Holt then did, he made a number of mutations in the putative pore of the channel in the surrounding and wanted to ask, do these mutations affect ion selectivity or conductance through the channel, which would really suggest that they must affect the pore of the channel. Somehow, all kinds of weird things pop up on my screen and I can't advance. Okay, sorry. Somehow, okay. Sorry, my presentation was stuck for reasons I don't know. So Jeff, he generated 17 mutants and showed that 11 out of the 17 mutants affected either conductance or calcium selectivity, providing strong evidence that this molecule is actually part of the channel pore. Now, what we were wondering actually, how does this TMIE molecule fit in? What you see here is the TMC model, and next to it, a molecular model of the TMIE molecule with its transmembrane domains and this helices. While TMIE is a small molecule, the helices actually in actual molecular dimensions relative to TMC are rather large. What Christopher Cunningham in my laboratory showed that TMIE binds very tightly to TMC1. Furthermore, he demonstrated when you have hair bundles in mice which express only TMC1 but no TMIE, we cannot observe transduction. And when we have hair bundles which express TMIE but no TMC1, we also don't have transduction, suggesting that interactions between these two molecules within the bundle are required to make an ion channel, functional ion channel. Now, interestingly, binding of TMIE to TMC1 is mediated by this helix here close to the leaflet of the cytoplasmic side of the membrane. And very strikingly, 3 mutations have been identified in humans which lead to deafness, and they're all localized within this helix. Now, we tested if these mutations individually were disruptions between TMC1 and TMIE, but Chris in my lab showed actually that that is not the case. These mutations do not act by disrupting the interaction as you can measure in biochemical experiments. So we then wanted to ask what do these mutations actually do and how do they act? So we generated by CRISPR-Cas strategies knock-in mice carrying individually these mutations and characterize them further. And what you can show, and that's shown here for one mutant, but it holds for all of them, when you record currents from these hair cells, you have here wild type and here a mutant, what you can see current is drastically reduced, which has in part to do with the fact that the transduction channel complex is not very stable. It falls apart. You lose transduction components from the bundle. However, you have significant current left. And that allowed us to do an experiment. Now, usually when we, to analyze ion channel properties, usually when we record from hair bundles, you see a diagram, we of course record from all the complexes, all the transduction complexes at each tip length shown here in this red. However, when you perfuse EGTA in the bundle, you can break the tip lengths until a single transduction unit is left behind. Now you can analyze the properties of the single transduction unit. And that's what Xu-Feng in my laboratory did. And what he showed actually, that there's severe changes in the properties of the channel. What you see here is single channel recordings from a control mouse, where you see here single channel events which are evoked by deflection of the bundle by a certain degree. From these curves, you can actually determine the conductance, the single channel conductance. Now, when we carried out this experiment with the point mutation, here shown some recordings for one of the mutants, and here's a quantification, we saw there was a 20% reduction in conductance, and we of the similar change for second mutant. We also measured the reversal potential, which tells us something about the calcium permeability of the channel, and was severely affected. Now, what this then shows that not only TMC affects the channel pore properties, but also does TMIE, suggesting that TMC1 and TMIE in some way assemble into a channel complex, that they're both constitutive component of this complex, which in some way influence ion flux. Now, what's peculiar that this TMIE domain, which affects ion flux, is not in the transmembrane segment, but cytoplasmically. And something I will return later to is here this model that one model here could be that the transmembrane domain of TMIE actually sits next to this cavity, which is exposed in TMC to the lipids, while TMIE sits very close here to the pore region. And one could imagine is that it works as a molecular gatekeeper, and that when you make mutations here, you affect the ion flux through the pore and also the selectivity, the calcium selectivity. Now, Xiaoping Yang in my laboratory showed this is not all what's in the channel complex, but there's two more molecules. And one of two you can choose, actually, which is there. This is a CIB2, CIB3 molecules. What Xiaoping showed that this molecules binds to a TMC1 TMC1 helix, which is buried in here and not really revealed by the structural model. And the CIB2-3 molecule binds very tightly into this complex, and there's one molecule per TMC1 molecule. Remarkably, Xiaoping solved the crystal structure of this molecule in complex with TMC1, and it looks in structure identical essentially to regulatory subunit of voltage-gated potassium channels. And again, a theme I will briefly return to later. Now, all this together then suggested an ion channel configuration, which is this, which consists of these 6 proteins, a dimer of TMC, a dimer of TMIE, and the CIB2 or CIB3 molecules. Remarkably, Eric Guo's lab recently purified the TMC ion channel complex from nematodes, and that is the structure they observed. And then they solved the structure by cryo-EM. What you see here is these 2 TMC molecules, just as predicted from David Corey's and Kenton Schwartz's work, where we have this dimer of TMC1 connected to 2 TMIE molecules, which co-purified with the complex, and the CALM1 molecule, which is the CIB2, CIB3 homolog of nematodes. So really showing that what I showed you before in a cartoon fashion is actually a true model of the ion channel complex. Now, what also the model predicts from Eric Guo's lab, similar to what David Corey and Jeff Holt have shown, that the ion channel pore sits somewhere at the side here of the channel, very close to a lipid-filled place in close proximity to TMAE. What we actually don't know is if this is open or closed conformation. It could be actually that this area undergoes significant structural changes when the complex is under tension and may very severely change this whole area in how the different helices arrange towards each other. So I think we need more work to get channel structures in various conformations, just as we have seen beautiful work in the previous talk for Prestin. And we also need, of course, to get the mammalian proteins. Now, these molecular studies then have brought us an outline of the mechanotransduction channel complex, or the whole machinery together with the tip link. But one question which is really still open is, how is the channel actually gated by force? When you look at this TMC proteins, TMIE and the CIB2, it's not inherently clear how mechanical force would open the channel. Of course, as I'm sure all aware of, the tip link has been implicated very strongly by work from David Corey and Jim Hudspeth most prominently in gating the transduction channel. But what is not clear if the channel is directly linked to the tip link, or if the tip link might deflect and deform the membrane, and that leads to opening of the channel. And that is something we have looked at. And there was actually an older paper which was published from Wei Chong in my laboratory before we even knew the molecular components of the channel. What Wei identified is this protein LHFPL5. My apology for the strange names, we didn't invent them. But this molecule has 4 transmembrane domains, and work from our laboratory and Robert Fettibla's laboratory showed that these molecules sits actually here at the lower end of tip links, binding both the channel and the tip link complex. When you have mutations in humans in LHFPL5, you will suffer from deafness, and the same is true in mice. We actually generated mouse lines with a mutation in LHFPL5, and you can see transduction currents were severely reduced, but not completely abolished. Now, there was something very peculiar when we analyzed these currents in more detail, that is shown here. Usually what you see is activation of the channel in wild type or in mutant. It's extremely fast. The activation time constant is below 10 microseconds. It's actually too fast to accurately measure. However, when we expand here the time scale somewhat, you see here the activation curve that we record in wild type, and it's not an accurate activation curve because of some limitations in our equipment. But what is clear when we record from the mutants, it's severely delayed. You see this here perhaps in a better representation at different open probabilities of the channel. In the wild type, the activation is below, it's in a few microseconds, while when we remove LHFPL5, it's within milliseconds. And that suggested to us a potential model that TMC1 might, and TMIE, this complex, might be linked to the tip link via LHFPL5, and that this molecule, when you take it away, the force coupling is reduced, leading to a delay in activation of the channel. And we wanted to test this model, and that is what Chu-Feng Qiu in my laboratory did. And he took Chu-Feng to the advantage of the fact that LHFPL5 is a member of a small family of 6 proteins. Now, what Chu-Feng asked is, what happens if you take hair cells from LHFPL5 mice and you rescue them? You stick in these different proteins and you ask, can they all rescue transduction? And what Chu-Feng actually showed us that among all the LHFP proteins, only LHFPL5 can rescue transduction. The mutant hair cells have no transduction or very little. None of these proteins can rescue transduction, even though they all can go into stereocilia. Only LHFPL5 can rescue transduction. What Chu-Feng also showed in collaboration with Chao Ping is that while L5 binds very tightly to the tip link and also to TMC, not shown here, other proteins which don't rescue transduction bind much weaker to these proteins. Now, then what Chu-Feng wanted to test if there's a good correlation between binding and function. And so what we did is we mapped binding sites, and that's summarized here. I don't have time to show you the data, but there's in the N-terminus of protocartier 15, there are binding sites for alpha, for this L5. I will just refer to these molecules for simplicity as L5. There are binding sites for L5 to the tip link, while binding sites for TMC1 are spread more throughout the transmembrane helices. Now, what Chu-Feng then asked is maybe the difference between other L proteins in L5 that the binding sites are not maintained. So what he did is he generated chimera, and he took L5 and its closest homolog L3, and he made various chimeras and asked which one of these chimeras are functional, and does function correlate to strength of binding. And that is shown here. L5 binds tightly to protocartier 15, L3 does not. Chimeras which have the N-terminal part of L5 bind tightly. Those which don't have the N-terminal parts do not bind tightly. It's a little more complicated for TMC1, where you need L5 binds tightly, L3 does not. The chimeras don't bind very well because the binding site is more spread. When we now took these different chimeras and asked we can rescue transduction, we saw there's a very nice correlation between binding and between function. And what you see here, maybe the right curves show it best and easiest. You see here a rescue if you have an L5 mutant hair cells and you stick in the wild type L5, you rescue currents. If you stick in the well-binding L5, L3 chimera, you rescue binding. But proteins which don't bind well don't rescue. And you see also this shift in the IX relations and the current displacement curve showing that you need more force to open the channel. So there is the force transfer is inhibited or reduced when you reduce binding strengths. And I'm showing you one example. We have made many mutations to confirm this finding, suggesting then that one function of L5 is to tightly link the tip link to the transduction channel. Not shown here, the channel, but we come to this in a minute, because there was one other finding which was really peculiar, and that is this N-terminal cytoplasmic domain of L5 highlighted here. So what Chu-Feng actually showed is that TMC1 and TMC2 interact through their transmembrane helices with multiple helices from L5, but this little cytoplasmic domain is very, very strongly bound by the N-terminal cytoplasmic domain of TMC1 too. And we were puzzling for a very long time what that matters, or what that means, and what the function of this is. What was very peculiar about is when the structure was solved recently of the nematode channel, it turned out that this small cytoplasmic domain has 3 helices. And one of these helices is remarkable. This helix H3 is an amphifathic helix, and amphifathic helixes are no strangers to the mechanotransduction field. You see here a number of mechanotransduction channels that have been purified and characterized over the years. And you see highlighted here in red these little helices. These helices are amphifathic helices, most prominently many of them here in the piezo mechanically gated ion channel. And these helices are critical for the gating of these channels by force. Now, one of these helices is found in the cytoplasmic domain here of TMC, and it binds to this L5 molecules. So we wondered, is this a critical gating element for the channel? And is this in some ways engaged by this linker between the tip link and the channel? And to test this model, we generated mutants in TMC1 molecule in these different helices. And then we asked, are they functional? We stuck these mutants into hair cells from TMC1, 2 mutant hair cells, which have no transduction. TMC1 itself can rescue transduction. Mutation in helix 1 or helix 2 can rescue transduction. But when you mutate helix 3, you completely abolish the mechanical sensitivity of the channel. We have many more data on this, which I don't have time to show you, but suffice to say, this helix is critical for mechanical gating of the channel. Now, peculiar, when we take the the L5 molecules, and we mutate this little cytoplasmic domain, and we actually substitute it with the cytoplasmic domain of one of its homologs, L3, and there's only 4 amino acid change, we generate an L5 molecule, which is no longer functional either in gating the channel. And actually, what I described to you before, the force transfer on the channel or the response of the channel to force is altered by these mutations. I'll show you 2 more data slides where we went one step further. Now, we took this little tail in L5, these mutations we actually put into mice. What you saw before were rescue experiments in explants. Here we made it with CRISPR, a genetic modification in the cytoplasmic domain of L5, where we converted these few amino acids into the amino acids, or here a deletion and a point mutation in L3. So we changed this little tail. And that is actually what I had shown you functionally here, this mutant, which in our explant didn't function very well. And then we analyzed these mice. The first thing we asked is when we expressed this as mice, the mice were deaf, and we asked, is the transduction complex normally assembled? What you see here, in a normal wild-type mouse, we can see tip-linked protein staining hair bundles that is completely normally maintained in the mutant, and we have done scanning EMs too. Tip-links are completely maintained. The L5 wild-type protein, of course, goes into the bundle. The mutant goes to bundle. Proteins like the TMC channel go to the bundle as well. We have looked at all components that we know. Everything looks normal in the channel, in the bundle. The morphology looks normal. The proteins are there. However, the mice are deaf, and when we record transduction current, there's very, very little current. Now, this here is, of course, the current that I showed you for the whole bundle. We also carried out, when we fused EGTR in the bundle, we can carry out single channel recordings. Single channel currents are hardly changed. What this then means that even though we have the full complement of channels in the bundle and we have tip-link there, the channels are less active. They cannot be fully activated by this very, very small mutation just in this L5 molecule, leading us then to suggest a model where this L5 proteins serves as a mechanical link between the tip link and the transduction channel complex. And we think that L5 is a molecular coupler which engages with the gating domain of the channel, which is conserved across mechanotransduction channels. And we think that TMIE might serve as a molecular gatekeeper. So when you apply mechanical force, this is propagated through the system. You pull up on this L5 molecule. This force is sensed here by this gating domain and likely leads to an opening movement in this gating swing, something we're currently actively testing. It would explain one phenomenon, that the pore of this channel is extremely big, but it would actually provide an explanation how in a very, very large pore, you can actually have a constriction near the end and a gatekeeper which helps to control the ion flux through the channel. Now, I want to finish with an analogy. When you look at the mechanotransduction machinery, we actually think we're looking at something like a molecular platypus. The platypus was introduced to British science in the late 1800s, and it's a very strange creature, and the anatomists didn't want to believe initially that this wasn't a joke. It was shipped from Australia. It's an egg-laying mammal. It can do electrolocation, which is unusual for mammals. It has venomes, which mammals don't have, a duck bill, a beaver tail, an otter-like webbed feet. Now, what's the analogy here to the transduction machinery? Well, we think evolution reconfigured players that we know from other molecular machines to build these new machines. Caterins usually form adherence junctions, but here it was reconfigured into a dimeric structure with a completely new adhesion interface, which David Corey showed. This is dimeric, it's completely reconfigured. TMC1 and TMIE are orthologs of other proteins or belong to the families which were in some ways repurposed to form this ion channel pore. L5 has homology to TARP proteins. These are regulatory subunits of glutamate receptors. So there they work in a ligand-dependent manner, but here they're reconfigured to work in a mechanical member. And finally, the CIB2-3 proteins, they look essentially identical in structure to voltage-gated regulatory subunits. So it's almost like this machinery has cobbled together components which had been used before, put them together in a new context to build this beautiful machine. And I think in the next talk we will hear much more about the intrinsic properties of the channel, which are important for gating for gating and how the channel responds to forces intrinsically. And with this, I want to finish, just acknowledge 4 of the people which have been instrumental to this work that I documented here: Chu, an electrophysiologist; Chris, a geneticist and biochemist; and JP and Xiaoping, which do structural and biochemical work. And I also want to thank all of you for your attention.
Thank you very much, Uli, for the very nice presentation about the mechanotransduction channel structure, the complex. There are many players, those which the homology to this platypus was really excellent. Any questions to the speaker from the audience? We are looking, Professor Kellermayer or anybody from the audience. I just would like to ask one very small question, because time is very sensitive now. Did you consider how the lipid composition would alter the gating or the coupling of these machineries, or are there any experiments related to that? Because
yes, this is actually excellent question. I left the lipid out of this, but the lipids are very clearly important. It's known, for example, that PIP2 clusters at the tips of stereocilia. It is known that PIP2 also binds to this TMIE molecule, which is part of the channel. And molecules like L5 are actually known in other contexts to form little lipid microdomains. And finally, the work from Tony Ritchie recently has shown that the viscoelastic properties play a role in regulating channel activity. So I do think lipids will be very, very important. They were left out of this model, and we really still have to probe how they work in this context.
Thank you very much. Any other comments or questions to the speaker? If not, then I would like to thank you very much for the participation, and also thank you very much for the presentation. And I would like to get it somehow to you, both the diploma and also the medal. Okay, and thank you very much with this. Your presentation, and I would like to call the next speaker. Thank you again. Thank you. I would like to call the next speaker, who is going to be David Corey, and he's from the Harvard Medical School, and he's going to talk about the mechanical gating of the TMC1 transduction channel in hair cells. David, please go ahead.
Thank you. I'll try sharing my screen. Sorry, wrong share. Yes, that's better.
Very good, thank you. This is the one, and we just have to see the presentation. Yes, very good.
Okay, so first, it's a great honor and pleasure to be asked to speak at the symposium honoring Georg von Békésy. Although he spent several decades at my institution, I never met him. We didn't overlap. He had moved on by the time I came. But he was tremendously influential when I was a graduate student. An interesting quotation from von Békésy is seen here, where he describes some of the challenges that are being faced. This is in 1962, after he had won the Nobel Prize, and it was a review article. But he notices a fundamental question is how the vibration forces eventually lead to the opening of an ion channel, how they cause electric current to flow into the cell. And he calls this the final mechanical transformer. And in this paper, he goes on to describe several kind of molecular rearrangements that might mediate such a transformer. And frankly, they're not really very representative of biological system, but you can see him kind of grasping for a sense of what this very important part of the hearing system must be. And so I want to tell you a little bit about some of our studies that have tried to get at what this molecular transformer is. And in doing so, I'll start by describing also some things that Uli went over, but perhaps expanding them in greater detail. And so one of the problems had been all along that we understood that the chip link was made of cadherin 23 and protocadherin 15, that there were other proteins that Uli had identified, especially through deafness mutations. But there had been many false starts in trying to understand what the actual ion channel was that was opened by these forces. Really seminal work by Jeff Holt, Gwen Jeliak, and Andy Griffith showed first that mice lacking both TMC1 and TMC2, a close homologue, never have functional hair cell transduction. And so that was a clue, but there are other proteins that are also essential. And Jeff went on to find a spontaneous mutation in TMC1 that alters the conductance of the hair cell transduction channel. And this led much more solidly to the idea that TMC1 was actually a pore-forming subunit of this complex. But a few spontaneous mutations wasn't really enough. This was still very controversial in the field. And so it was really important to be able to make further mutations, but just knowing the sequence wasn't good enough. We really needed to have a way of rationally designing mutations. We needed something based on the structure. So early on, we decided to try to figure out what the structure of TMC might be. One of the first was just to try to understand the multimerization of TMC1. And we found, for instance, with fluorescent size exclusion chromatography, that TMC1 runs at about a molecular weight of somewhere around 260 kilodaltons, whereas the monomer is closer to 80 kilodaltons. So that suggested that it was assembling as a multimer of 2 or 3 subunits. If we use cross-linking of native protein, we found that in the absence of a cross-linking agent, it ran at a monomeric weight, but in the presence of this cross-linking agent, DSBU, it seemed to run as a dimer, but we didn't see evidence for higher multimerization. In native gels that are non-reducing and non-denaturing, again, the TMC, the purified TMC, seemed to run as a dimer. And if we use size exclusion chromatography with multi-angle light scattering, you could see that where the protein elutes, there's a multi-angle light scattering signal of the protein and the surfactant. But if we subtract the surfactant, we can see that the protein runs at a molecular weight that is equivalent to a dimer. We also tried to get an idea of what the structure would be, and we, as many others did, attempted to get a cryo-EM structure for TMC1. We never got that far with the cryo-EM structure, and most groups didn't either, but it was clear from early images that there was a twofold symmetry in the cryo-EM structure, and you can see in many of these a sort of a twofold symmetry. So all of these led to the idea that TMC1 assembles as a dimer. And this was really peculiar because almost all the ion channels that we think about, the ones that are in textbooks, are made by different subunits coming together in 3, 4, 5, or even 6 different subunits to form a central pore. But it wasn't clear how you could put 2 together to form a central pore. And so this was seeming to be a very strange kind of an ion channel. At the same time, there had been other dimeric ion channels that had been described, especially work from Chris Miller's group on the CLC channels showed that these were dimers, and work on the TMEM16 channels from a variety of groups showed that these were also dimers. And wonderfully, there were either X-ray crystal structures or cryo-EM structures of both of these. So we could begin to see whether TMC1 was anything like these known channels, these known dimeric channels with known structures. A very quick way of looking is to try to look at the transmembrane prediction. And if a lot of different algorithms for predicting transmembrane segments gave differing results for TMC1, which was one of the early challenges. It wasn't clear whether there were 6 or 8 or even 10 transmembrane domains. But whatever it was, it really wasn't like the CLC channels that had 14 transmembrane domains. On the other hand, if you compared TMC1 to the TMEM16 kind of low-scale architecture, it did seem to match. In particular, there were questions about whether this hydrophobicity region represented 1 or 2 transmembrane domains. And there was kind of a varying pattern in TMC1 that was like that in the TMEM16. These clearly were 2 transmembrane domains. And so we had a pretty good one-to-one matchup between the TMC1 sequence and that of TMEM16. And then that allowed us to kind of align these. And others had shown that there was at least some amino acid similarity between the 2 groups of ion channels, not much, but especially in the 7th, 8th, 9th, and somewhat in the 10th transmembrane domain, there was an amino acid similarity between various TMC1s from different species and different TMEM16s. And that allowed us then to plot a phylogeny tree. And you can see here, for instance, the human TMC1 is distantly related to the TMEM16. It's fairly far apart in evolutionary distance, but it's not all that much further apart than, say, TMC1 is from other members of the TMC family. And then Medrano Soto at about the same time showed evidence that not only was TMEM16 part of a superfamily with TMC, but there were 5 other branches, including the CSC branch, which we now know includes a channel known as OSCA, which is also a mechanically gated channel and has a structure also like the TMEM16. So this made it possible then to thread the sequence of TMC onto the structure of TMEM16. And remarkably, we knew from the structure of TMEM16 that there was not a single conduction pathway through the middle where the 2 subunits came together, but instead each of the monomers had its own conduction pathway, which seemed to be near the outside, near where the lipid approaches the protein. And in fact, one side of this sort of the proposed conduction pathway was exposed to the lipid, and might actually face the lipid unless there's, say, a 4th wall that's created by another protein. So this then gave us an idea of where the conduction pathway might be and allowed us to make rational design of mutations that would test whether the structure really made sense. And you can see here that the 4th, 5th, 6th, and 7th transmembrane domain create kind of a groove that is the hypothetical conduction pathway. And one question is, just from molecular modeling, can water and cations go through this groove? So we worked together with Marcos Sotomayor, and he first did molecular dynamics simulations to ask, could water, here represented in red, actually hydrate this groove? And you can see here that it definitely does. So this could be an aqueous path for ions to go through from the outside to the inside. And he went on to do molecular dynamics simulations with the, let's see if I can get this started. I'm going to This is so much fun. I want to try to get this going and take away the laser pointer. And now you can see that potassium ion represented in red can diffuse along this groove, and that it's quite happy in this in the region of the groove. And Marcos went on to show with further molecular dynamics simulations that one after another potassium ions can pass through the groove, indicating that this is quite likely a conduction pathway. So we then were able to make a number of mutations in the TMC1 protein that are represented by the green residues. And in each case, we changed them to a cysteine. And not all of them were made at the same time, of course. One after another, we made cysteine substitutions for the regions of TMC1 that we felt were likely to line the pore. And then there were a few mutations that were further away from the proposed pore domain. And then we collaborated with Jeff Holt to test these mutations in intact hair cells. So one of the big problems is these proteins really don't work in an expression system. And so the only way to really test them is to put them back into a functional hair cell. So the strategy is one by one to make a single mutation in TMC1, package it in an adeno-associated virus vector, inject that into the inner ear of a mouse, but specifically a mouse that's lacking both TMC1 and TMC2, so that the only TMC that's expressed in the inner ear is one with a specific mutation that we introduced. And then after 5 or 10 days, the cochlea can be taken out and studied with patch clamp recording and direct mechanical stimulation of a single hair bundle. And this is work that Bifeng Pan and Jeff Holt did. Now, there are a lot of ways that you can mess up the function of a protein. You could, for instance, make a mutation in which it fails to even get to its normal site of action. And one of the wonderful things about a cysteine mutation is that often the cysteine substitution doesn't change the channel properties, but in a time scale of seconds, we can change the cysteine side chain to something that's much bulkier, particularly, for instance, if we add the MTSET reagent, then you get a covalent modification of the side chain that adds this bulky group, and this might be much more likely to change the channel properties. And thus Bifeng would be stimulating a bundle, and as the individual deflections of a bundle caused individual pulses of current that were measured by the patch pipette, the MTSET was applied to the bundle to make an acute mutation on a time scale of seconds. Now, if you do this to a wild type hair cell, with each of these mechanical stimuli, perhaps 3 per second, eliciting an inward current, then it turns out that the actual application of the MTSET does not change the channel conductance, and you see that there's no change in the amplitude of the current over a time scale of about 10 seconds. But for many of the mutations circled here, you can see that within even tenths of a second, there is a marked reduction in the amount of current coming into the hair cell. And some of these modifications are extremely quickly. So we know the channel is there, we know that it's functioning, and now we can very acutely modify a specific aspect of the channel function. And I won't go into all the detail, but it happened that many of the mutations shown in magenta changed the conductance and also the ion selectivity. Others of them only changed the ionic selectivity, the selectivity between, for instance, sodium and calcium. One was really nasty. It changed the current even without the MTSET, but others that were further away from the putative conduction pathway did not have any effect. So this gave very strong evidence that this is, in fact, the pore of the TMC1 ion channel. So first, it tells us that TMC1 and TMC2 are definitely pore-forming subunits of the mechanotransduction complex, and this pretty much answered a question that had been longstanding for decades. there's this peculiar arrangement where we have two distinct pores in one dimer. We think that they're independently gated, but it's possible that they're cooperatively gated so that if one opens, the other one might be more likely to open. As I mentioned, the pores face the lipid on the outside, unless perhaps another protein subunit forms the fourth wall of the channel. And as in the models that Uli showed, it's attractive to imagine that each monomer binds one protocadherin of the dimeric protocadherin tip link complex. And try to get my pointer back at this point. Never mind. And finally, it's really important that the suggested transmembrane arrangement then provides other testable hypotheses for how this might work. Okay, let's see whether that works. Good. But the question still is, where's the gate? I mean, how is it that this pore could open or close? And for that, we turn to rapidly developing protein prediction models that were developed in a number of different laboratories. So here are three structures. One, the homology structure that we generated by homology to the TMEM16 structure. Another two are TR Rosetta and Rosetta Fold, developed in David Baker's lab. And you can see that there's really not much difference between the structure proposed from a fairly distant homologue and that predicted by these de novo protein structure prediction programs. And then even more recently, the AlphaFold 2 suite of programs came out. And you can see that they too predict something that's very much like the original homology model prediction. And AlphaFold 2 can be run with or without reference to other existing structures in the database. And it turns out that whether you run it with or without access to other structures gives us essentially the same structure. So we now have 5 things that are all essentially giving us the same structure, giving us not certainty, but a lot of confidence that these protein structure prediction programs can tell us something about the way the protein might be organized. And now, most recently, the C. elegans TMC protein has been solved by cryo-electron microscopy. And here we compare the AlphaFold 2 structure in green of a human TMC1 to the C. elegans TMC. And these two proteins have been separated by 400 or 500 million years of evolution, but they still have essentially the same structure within 5 or in some cases 10 angstrom. That also gives us a lot of confidence. So we went on then to ask, what if we run the AlphaFold program and try several different iterations? And we ran it, for instance, 20 times with different seed values. And the remarkable thing was that in 16 out of the 20 times, you can see here in gray, the 4th transmembrane domain comes very close to the 6th transmembrane domain. And you would think that it might actually occlude ion conduction through the channel. But in the other 20% of the runs, the 4th transmembrane domain and the 3rd transmembrane domain are shifted over to the left, and that seems to open the pore. So it's possible that one of these structures represents a closed conformation of the channel, and the other structure represents an open conformation of the channel. If we look at these structures in more detail and use, for instance, the holes program to look at whether there's an aqueous, that is an access of water molecules to the pore, in the left structure, water really can't get in there. But in the right structure, you can see that there is a complete aqueous pore all the way down this groove. So you could imagine that these two then represent an open and a closed conformation, and that, let me see if I can get this to go, and that the actual gating movement is represented something like this movie, where the 2nd and the third and fourth transmembrane domain move away from the sixth transmembrane domain to open and close the pore. And you can see that there are other molecular rearrangements that are happening at the same time that may be linked, for instance, to some of the protein connections that Mueller described. So we decided to see whether we could test what the involvement of the fourth and sixth transmembrane domain in the gating. And for this, we made just a few mutations. And in most cases, rather than changing these to cysteine, we simply mutated them to an alanine. But we reasoned that glycines in the transmembrane domains would confer flexibility to the transmembrane domain. And if we mutated the glycines, they might be less able to move, and the channel might have more difficulty gating. So we first changed this glycine at position 411 to an alanine. And again, one by one, we made the mutations, put them into an AAV vector, injected it into a TMC knockout mouse, and then looked at the physiology after 5 or 10 days. And here we found that if we mutated this glycine, well, first we checked whether the introduction of a mutant TMC might fundamentally affect the behavior of the hair cells. So we started by simply introducing the wild-type TMC. And you can see that if we introduce the coding sequence for the wild-type TMC into a mouse lacking its normal TMC 1 and 2, we still get currents. And the activation curve, the relationship between displacement of the bundle or deflection of the bundle and the open probability, is essentially unchanged from a mouse that is normally expressing its own TMC. Similarly, the single channel current that's measured by non-stationary noise analysis is essentially that the same as that previously measured by Jeff Holt in a similar preparation. Now, if we make the mutation, again, this glycine at kind of a hinge in the TMC4, we find that the activation curve is fundamentally changed, that the curve is broadened, and so it takes a larger displacement of the tip of the bundle to cause the same increase in open probability. And this, it's a little bit hard to know how to think about this. In a very basic model that is by now about 40 years old, the shape of the activation curve was really determined by just two parameters. One is the stiffness of this hypothetical gating spring, and the other is the distance that the ion channel moves when it opens. And if the curve is flattened, as we saw here in this mutation, it might be that the gating spring became softer, or it could be that the channel didn't move as far when it opened. Now, we think it's probably moving as far, at least based, for instance, on the single channel conductance. And that is the conductance is not significantly different from the wild type. So we think that the pore is opening as much as it could. And I think we really have to start thinking a little bit in more complex terms about what the activation curve actually represents. For instance, if force is conveyed from protocadherin 15, perhaps through LHFPL5, and perhaps through the protein, then at any stage, if that force is less effectively conveyed to the parts of the protein that undergo the gating, that might also change the slope of the curve. So perhaps this is a failure of the tight linkage between a force generated by bundle deflection and that actually opens the channel. We made other mutations. For instance, we changed glycines in the sixth transmembrane. And here, we didn't see a change in the slope of the activation curve. But in all three cases, we saw a shift of the activation curve. And this would represent the channel being less likely to be open. That is, these mutations seem to stabilize or favor the closed state of the channel. It's not exactly clear how we would be stabilizing the closed state, but it does suggest that these two glycines are involved somehow in the gating process. And again, there's not much change in the single channel conductance for these mutations. And then another mutation is one for this aspartate at position 528, and this is right in the center of the conduction pathway. So for instance, if we change the aspartate to an asparagine, then that substantially reduces the charge that would be facing the pore. And we find in this case that indeed the single channel conductance is hugely decreased, which is not surprising. But this mutation and other mutations of aspartate at 528 also tend to change the activation curve in not always predictable ways, but at least we can say that this seems to be involved in the gating in some sense. So we can say that mutations that reduce the flexibility in the fourth and sixth transmembrane domains, that is here and here, do affect the gating. Mutations that change the charge, and there are others that I haven't showed you that change the hydrophobicity of TM6 also affect the gating. And so they're consistent, at least, with the idea that these are very important parts of the gating movement. But at the same time, there's a lot more work to do in testing the predictions of these structures, for instance, in trying to figure out whether this large movement of the third and fourth transmembrane domain is more than just an AlphaFold prediction. So at this point, I just want to mention the people in my lab, especially those highlighted here, who've done a lot of the work. The early work was a wonderful collaboration with Jeff Holt and Bifeng Pan, and we continue to enjoy a collaboration with Marcos Sotomayor on both the gating and structure of the mechanotransduction complex. So at that point, I could take questions.
So I would like to ask first the audience if there are any questions to the speaker. A short one question from the floor. If yes, there's one question from Tibor.
Yeah, thanks for the lecture. If I recognized it correctly, you measured the current of the channel at P6, at the age of P6 of the mice. Do you think, or can you imagine, that maturation influences the property of the channel, the surroundings, or you believe that this is rather the finite, the mature property of the channel?
Well, it's a little bit hard to know, and I should point out one thing, which is that early in the postnatal life of the mouse, the TMC2 channel seems to be the main contributor to the channel. In our experiments, that has been genetically removed, so we're only looking at TMC1. And these are not experiments that we did, but Bifeng Pan and Jeff Holt have studied the properties of the channel from about postnatal day 5 up to about postnatal day 14 or 15, and don't see a substantial change in that amount of time. But we don't really know beyond that. It's very difficult to do these experiments at an age of, say, P30 or P60. So I can't really say whether this represents a mature property or characteristic of the channel.
Okay, thank you very much. Any other questions? If not, then we would like to thank very much again David for the presentation, and I would like to I will make sure that this will get to you. Okay, thank you very much indeed for the contribution. Thanks.
Thank you very much.
Okay, so thank you. And I would like to ask the last speaker before the break to join us, Jim Hospes. He's from the Rockefeller University, and he's going to talk about critical behavior in the active processes of the cochlear hair cells. And he's going to join us on Zoom as well. So, Jim.
Thank you very much. I'll start out by trying to share my screen. Is that working correctly?
Yes, so far so good. And with the presentation view, we will, yes, works very well. Thank you.
So I wish to continue an analysis of some of the issues that have already been brought up. And in particular, one of the major issues that has stemmed from the earlier work of von Békésy. And that is, he showed very elegantly that how the basilar membrane of the cochlea resolves different frequencies of stimulation into independent mechanical responses. And we now have a view of this process, something of the sort shown here, in which the motion of the basilar membrane is fairly tightly confined to a relatively short distance along its length, but is passive. The basilar membrane is passive beyond that point and has relatively small amounts of motion before. This is in contrast with the original finding. Von Békésy originally worked, as has been mentioned, with cadaver specimens. In that case, the curves were quite broad. There was frictional damping from motion through the liquid and not sharp tuning. Now we see much sharper curves, and this has led to the idea that there's some sort of an active amplification, an active process that enhances the motion of the basilar membrane and sharpens frequency selectivity. And a major question is, what exactly is that active process? The active process has 4 characteristic properties that were pointed out first by Jeff Manley and always occur together throughout the vertebrates, whether we're talking about amphibians, reptiles, including birds, or mammals. First, there's amplification. So our sensitivity is enhanced by something like 100-fold to 1,000-fold over what would be the case in a passive cochlea. We can therefore detect motions down to a fraction of a nanometer of internal motions in the ear. Frequency selectivity is very high, so we can detect frequency differences that are a very small fraction of 1%. There's compressive nonlinearity. So a 1,000,000-fold range in stimulus amplitudes, which represents a 1,000,000,000,000-fold range in stimulus power, is somehow telescoped into just about 2 orders of magnitude of output. And finally, in most normal ears, there is spontaneous otoacoustic emission. In a suitably quiet environment, the ear spontaneously emits 1 or more frequencies of sound. One can see these same properties by looking at the behavior of the basilar membrane. This set of schematic diagrams represents basilar membrane responses on a logarithmic scale as a function of frequency. So in every case, there is some particular best frequency at which the response is best. And if the cochlea is de-energized or the animal dies, the response falls to this lower level. It's only 100th to 1,000th as big as it is normally, indicative of the active amplification. Next, a de-energized cochlea, even if it is exposed to far louder sound, has very broad frequency tuning, whereas a normal cochlea has quite sharp frequency tuning. Next, we have compressive nonlinearity. If one increases the intensity of sound 10 times or 100 times, away from the characteristic frequency, we see a linear response. The response gets equally bigger. But at the characteristic frequency, the response grows more slowly, more like the cube root of 10-fold, and that allows additional lower levels of sound to be heard, basically telescoped in below those shown here. And finally, as I've said, we have spontaneous otoacoustic emissions in which various pure tones are emitted, anywhere from 1 to more than 30 in perfectly normal ears. So obviously this active activity is occurring in the cochlea, and it's coming from hair cells. And Péter Dallos first gave really important experiments that showed that the outer hair cells seem to be prominently involved in the process. How the outer hair cells do this, I think, remains a mystery. We've heard a lot about electromotility, but after 37 years of electromotility, we still don't know what it's doing. We don't know whether it actually enhances motion of the basilar membrane and of the components of the organ of Corti on a cycle-by-cycle basis. It might instead inhibit that motion. It might, in fact, dissipate energy to keep the cochlea from being damaged by very loud sounds. Or it might have a regulatory effect, a low-frequency static effect that tunes the cochlea or regulates its response to efferent stimuli, and by that means regulates its sensitivity. Basically, an outer hair cell has 2 lives. As we've already heard, when an outer hair cell's bundle is stimulated, cations run in, depolarization causes rearrangement of Prestin that shortens the outer hair cell and can, in principle, do mechanical work. But at the same time, the entry of ions, particularly calcium, can cause amplification of the mechanical stimulus at the hair bundle itself. And I think that that has to be taken very seriously as a candidate for the basis of the active process. In particular, there are a lot of hair bundles. In the roughly half-octave space where most of the apparent amplification occurs, there's something on the order of 200 or 300 outer hair cell bundles that can all contribute mechanical forces that could be involved in the amplification process. So I want to turn to how those hair bundles do mechanical work in non-mammalian cases, and then end by a small amount of information suggesting a similar process might work in mammals as well. We've done a lot of work using the hair bundles of the frog. They're large, they're sort of archetypical in their structure. They're relatively easy to investigate. And in particular, we can place a glass probe in contact with the tip of the hair bundle. And by that means, we can apply forces to the bundle. We can move the base of the probe. That pulls the hair bundle in the same direction. And because the glass fiber can be flexible, we can also measure how much it bends. And by knowing its stiffness, we know what forces the hair bundle is producing on the probe and vice versa. And using that type of apparatus, we are able to see all four qualities associated with the active process in individual hair bundles. So first of all, amplification. If we place a fiber in contact with the hair bundle and then begin to oscillate it back and forth, we find that the hair bundle is initially jittering around randomly, but it now locks onto the stimulus very nicely and it amplifies. The output of this hair bundle is about twice as big as the input. Next, the system is tuned in terms of frequency. These are low frequency hair bundles in a frog, so they're tuned down just at 10 hertz in this case. But you can see there's a strong response to that particular frequency and a lesser response to a lower or a higher frequency. If in fact the cell loses its active process through protracted stimulation, the response falls down to this passive level. Next, there is compressive nonlinearity. If we measure the sensitivity, how far the hair bundle moves as a function of applied force, we find that the sensitivity is very high for small stimuli and falls progressively and steeply as the size of the stimulus gets bigger and bigger. And again, an injured hair bundle reverts to this basal state without any active process. Those data are even more striking on logarithmic coordinates. So here we see the input, the base displacement of a fiber on log coordinates, and the output, which is the mechanical response of the hair bundle, again in a log coordinate. And we see a characteristic slope of about 1/3. And this is further seen or accentuated by measuring the sensitivity, how far the hair bundle moves as a function of force. We see a slope of -2/3. So the system is strikingly nonlinear. And the nonlinearity is suggestive of a particular type of mathematical object called a Hopf bifurcation. Now, a bifurcation is a qualitative change in the behavior of a dynamical system in response to a continuous quantitative change in some input. In other words, you turn some dial, and all of a sudden the system leaps from one type of behavior to a very distinct type of behavior. I won't go through the mathematics of this, but very briefly, the equation for a Hopf bifurcation says that the velocity of motion of the hair bundle, or a related parameter, is dependent upon real and complex coefficients times the position of the bundle. And then there's this peculiar cubic term, and finally the forcing that's applied externally. But what's striking in this system is if you poise the system near the bifurcation, where the parameter mu is near 0, and if you poise it, stimulate it with the right frequency, you find that the response grows as the 1/3 power of the stimulus that's applied, exactly the sort of phenomenon that I showed you. The sensitivity falls as the 2/3 power, again, the response I showed. The other thing about such a system is if the parameter mu is 0, the system is quiescent. It does nothing at all. But if mu becomes positive, there's a crossing of the bifurcation. The system now begins to oscillate spontaneously in a limit cycle. And this could well be the sort of thing that underlies the spontaneous otoacoustic emissions that routinely come out of an ear. So an individual hair bundle can recapitulate all four of the properties associated with the active process, including the active processes of mammals. Here's a demonstration of that last process, spontaneous motion of an individual hair bundle, now seen from above in a video. And these are measurements made with the probes showing that different hair bundles oscillate at different frequencies, high or low. They can be asymmetrical. They can cause various types of complex oscillations. And I think Dolores may talk more about this in a subsequent presentation. Where do these oscillations come from? How does this system become unstable, and how might it produce force? An ordinary elastic solid follows Hooke's law. There's a linear relation between displacement and force, as shown by the blue line. But if we make measurements of active hair bundles, we find that they have this extraordinary property of having a region of negative slope stiffness. There's a linear relation. There is a linear relation here, but in this region, the relationship is curved in the opposite direction. The system is unstable and has a negative stiffness. This comes about as a result of the activity of the mechanical channels. So here on the left, we have three pictures of an oscillating hair bundle. And then we abruptly spray onto the bundle gentamicin, which is a compound that blocks the transduction channels, apparently in their open state. Then when the gentamicin is removed, it diffuses away and the cells begin to oscillate again. What you can see is before and after the gentamicin, there is this region of negative stiffness. But while the gentamicin is present and the channels can't open and close, the system is entirely linear. It's Hookean. What we think is happening is this. Imagine that you have ion channels connected to a gate that can swing open through a distance d and is connected to a spring of stiffness kappa. Here are three of those channels side by side. If we apply a force, that force is equally distributed among the three channels. But if one channel opens, the associated spring relaxes. That means there's more force applied to the other two springs, and it's likely that another channel will open. And if that occurs, then the third one may open as well. So this system tends to be bistable. It's happy in the absence of force in the closed state, but it tends to show an avalanche behavior to the open state when a suitable force is applied. The consequence is that such a cell, such a hair bundle, cannot be stable at a position here in the middle. Throughout this red regime, it will always jump spontaneously either to this fixed point or to that fixed point. And this kind of behavior can be harnessed with a suitable energy source to make an amplifier or to make an oscillator. In brief, if you push the system into the red zone, it will tend to jump to the other side. Whether you push one way or the other, it can readily oscillate back and forth spontaneously, or it can be used as an amplifier because a weak stimulus will cause it to make these large jumps, by that means amplifying the input. We don't know at molecular detail how this comes about, but I think you've heard in the last couple of talks what could be going on. David Corey and I, and subsequently a lot of work by Pascal Martin, showed that there is an adaptation process that's driven by calcium ion. Calcium ion flows in through open transduction channels and then somehow interacts inside the cell to reclose the channels. We had no idea when we first proposed this model what the interaction might be, but the protein CYP2, which you've heard about recently, is now a striking molecular candidate to be the site at which this reclosure occurs. As you can appreciate, when this reclosure happens, there's force applied to these tip links that tends to pull the hair bundle back in the negative direction. So we have a little machine which we can push in the positive direction, but which a little while later will snap back in the negative direction. And such a machine can be harvested to do work on a cycle-by-cycle basis. So let's suppose we push the hair bundle first in the positive direction, the green arrow, and then back in the negative direction. During each cycle, channels will open and calcium will come in. Calcium will bind internally. That will do work, pulling the hair bundle back in the negative direction. After the channel has gone back to the negative direction, it's able to lose the calcium, to reopen, and again to undergo cycles of such oscillation. And in each cycle, some work is being done. It's work that's done by the binding of calcium, and it's powered by the ionic gradient that forces calcium into the cell. Such a model has the right properties. If we give a mechanical stimulus and look at the passive properties of the hair bundle, we would expect a very small displacement. If we apply the same force to a bundle in which the channels can open and close, but calcium is not binding, we get a slightly larger response. But if calcium can bind and add energy to the system, we get something very like what's observed, namely 100-fold amplification and actually a finite rise and fall time of the system. Moreover, the system is very highly tuned. So for a particular set of parameter values, there'll be one characteristic frequency at which it's most sensitive, and it shows compressive nonlinearity. The sensitivity or gain is greatest for a very small stimulus, and as the stimulus gets larger and larger, the gain gets smaller and smaller until it approaches the passive level. And finally, the system can go unstable. It can emit spontaneous oscillations. So here are the same inputs given 3 times as we change the control parameter. That was mu in the equation that I showed you. And as the control parameters change, approaching 0, the response to the same stimulus gets bigger and bigger until we traverse the Hopf bifurcation, and the stimulus now, without any stimulus now, we see spontaneous limit cycle oscillations, which might represent spontaneous otoacoustic emissions. Now we've tested the predictions of this sort of theory using this mechanical load clamp. We again apply a fiber to the tip of the hair bundle. We measure where the fiber is, and we use a piezoelectric system to move the fiber's base back and forth. But now we use a computer to specify whatever forces we want to apply through a dynamic command signal. And basically, we can fool the cell into thinking that it's experiencing a certain force, a certain elasticity, a certain mass, a certain drag, whatever we want. We have predictions based on a model that follow as shown here. If we look at the stiffness of the fiber as one control parameter and the force produced by that same fiber as another, we expect to have this fish-shaped locus in which the hair bundle will oscillate spontaneously. Outside that, there's a Hopf bifurcation, the pink line, and the hair bundle becomes stable out in this region. Moreover, we expect as we move towards the head of the fish, the frequency should get greater and greater. If we move towards the tail, the amplitude should become larger and larger. In fact, that's what we observe. This represents, again, the elastic load on a given hair bundle and a constant force. The black region represents that region in which there is no spontaneous oscillation. So we have this vaguely fish-shaped locus in which oscillation does occur. And indeed, as we move towards the head of the fish, the amplitude gets smaller. As we move towards the tail, it gets larger. And as predicted, as we move towards the head of the fish, the frequency goes up. As we move towards the tail, the frequency goes down. And these 4 points are represented by the records down here with some significant spontaneous oscillation. At this point, the motion is indistinguishable from thermal noise. The system is most effective when we're operating near the head of the fish. At that point, we have a strong response, a high quality factor that is sharp tuning. If we move out of this bifurcation region towards the quiescent region, in other words, beyond the fish, we get a smaller and smaller response until we reach this dotted gray line, which is the passive response. So we think this is going on all the time. We think if we're in a loud environment, the system resets itself to operate here outside the fish because no amplification is needed. If we're in an environment such as we are now, there's a modest amount of amplification. But if we go into a really quiet environment, if you're in your room alone at night and you can hear a pin drop, we're right around the bifurcation. The system is just bordering on, it's quivering at the boundary of instability. And if you move into a sound chamber, the system becomes profoundly unstable and begins to oscillate spontaneously. So what about the cochlea? The problem with analyzing these same issues in the cochlea is, again, what's shown in this diagram. Namely, various portions of the cochlea are doing different things at any given time. Here, things are relatively quiescent. Here, they're relatively quiescent. But in the active zone, some regions are moving upwards, some regions are moving downwards. The wavelength is changing. The velocity of propagation is changing. Everything is changing. So we've been interested in trying to produce a model system in which we can study the properties of cochlear hair cells in a more or less intact preparation without having the complexity of the traveling wave. We do this by isolating one segment, the middle turn of the cochlea, from the gerbil. That region encodes frequencies around 2 to 4 kilohertz. We can go somewhat higher, we can go somewhat lower. But the tonotopic map is well known in this case, and there are multiple lines of evidence that we are isolating things in which the cochlear activity should occur at roughly these frequencies. We isolate this segment and put it in a disc, I should say glue it to a disc, so it separates endolymph and perilymph. We can apply a potential across it to mimic the endocochlear potential. We can see it quite nicely in a light microscope. We can see the hair bundles of individual inner hair cells, the hair bundles of individual outer hair cells. And most recently, we can also use optical coherence tomography, which gives one quite a good view of the organ of Cordy, including the outer hair cells, diader cells, inner hair cells, and so on, and the overlying tectorial membrane. But more importantly, this allows one to measure oscillations of any of these visible components with the precision of less than a nanometer. And I should say our work in this regard is still at an early stage, but we're beginning to get reasonable results at this point, with some help from Marcel van der Heijden, who's visiting us right now. But I want to close just by showing you one record, which is an electrical record, the microphonic record from such a preparation. As we turn up the sound stimulus, the acoustic stimulus we use to drive this system, we find that there is a transition from a slope of 1/3, that characteristic slope that we saw in individual hair bundles and that is characteristic of a Hopf bifurcation, to linearity, which again is the norm at higher levels, both for individual hair bundles or in fact in human psychophysics. So we think that the same principles are occurring in these isolated cochlear segments, namely that we're operating at low sound intensities in a critical regime, taking advantage of the Hopf bifurcation. And when we get to some level near 80 decibels, the system begins to saturate because that system can no longer operate. So I'll close by saying, what are the advantages of operating at a Hopf bifurcation? First of all, they're the features associated with the active process. There's amplification of 100 to 1,000-fold. There's frequency selectivity of a fraction of 1%, and there's compressive nonlinearity over a 1,000,000-fold amplitude range. The other thing that occurs is an epiphenomenon, namely spontaneous otoacoustic emissions, which are not occurring in ordinary everyday life, but occur only in very quiet environments. There's another epiphenomenon that falls out of the same Hopf bifurcation, which is that one should hear these distortion products, 2F1 minus F2. Again, it's well known that humans can do this. Some composers have even used this in order to produce music in which no instrument plays the melody, but it's heard. In fact, György Ligeti, who had a partly Hungarian background, is among the prominent people who've done this. This type of bifurcation gives relative simplicity. It just needs 2 parameters. It doesn't require a lot, but just 2 parameters will produce resonant tuning and oscillation. A single parameter can cause it to cross the bifurcation. The bifurcation is local. That is, it occurs near a fixed point. So you can go from something that is amplifying but stable to something with spontaneous oscillation just by a minuscule change. The system entrains readily to stimulate, which is something you want hearing to do. And again, Dolores will talk about that in greater detail. And finally, it shows frequency constancy. As you move from a driven system that's highly sensitive to one that's spontaneously oscillating, the frequency does not change very significantly. And that's valuable because you want a system in which soft or loud sounds will be heard as the same frequency, rather than having the frequency change significantly as a function of the sound level. So I will stop there. I want to thank you all for your attention. And again, I'd like to thank the organizers very much for making this possible. Thank you.
Thank you very much, Jim, for the talk. And I would like to ask if there is any question from the audience to the speaker. Yes, Miklós.
Yes. Thank you very much for sharing this fantastic lecture with us. I have just two very quick questions. What is the role of the elasticity and the compliance of the tip link, and the role of the myosin 7 motor at the other end of the tip link?
Okay, so first, the elasticity of the tip link, we believe is important in what David Corey mentioned as the gating spring. I just happen to have a channel here. The channel has a gate, as you can see, and it's relatively non-selective, as you heard. The gate could be open or closed, and there's a tip link. So the elasticity of this cadherin 23 and protocadherin, allows a force at its other end to store a certain amount of energy, which can then stochastically open or close the channel. And we believe that that's what's going on in normal hearing, that as stereocilia move back and forth, the channel is opening and closing as a result of force transmitted through this spring. Now, as far as the motor is concerned, we believe that the motor is doing something at a low frequency, which is to bias the system into its active range. Suppose that this, again, is the channel. This is one stereocilium here. This is another one here. What I'm saying is when you listen to sound, as your stereocilia move back and forth, the channel opens and closes. There's a critical range of forces in which the channel can do this. That range of forces is the range in which it can amplify. If you're way over here, the channels are all open. They can't do anything else. If you're way over here, the channels are all closed. They can't do anything else. So the motor can set the right tension. The motor can move up the stereocilium until it's just at the point of criticality, where the thing is right on the borderline of oscillation. In the frog, the evidence favors as a motor a myosin called myosin 1C. It may well be myosin 7A in the mammalian cocktail.
Thank you very much. Thank you.
Thank you very much. Any other questions from the audience? And just to make it very clear, this was not a preset question, and then you had the demonstration tools right in front of you. So that's something that I'm pretty sure that's coming up very frequently, and then you were very well prepared. I would like to thank you very much again, and the diploma and the medal, and we will let you, we will make sure that you will get it very soon. And with that one, I would like to thank you for the talk and congratulate again.
Thank you very much. Thank you both.